Hello, my name is Hallie, and welcome to this assessment of vision. In this review, we will identify how the anatomy of the eye is able to absorb light from our environment and translate it into a nerve signal. Then we'll also identify how the brain is able to process that signal and allow us to do basic tasks like identifying the object in a picture. And by completing this review, you'll have a better understanding of the anatomy of the eye, as well as being able to identify the key functions of each structure. And you'll also be able to map out the visual processing pathway, which means that you'll understand how visual information will move from the retina to the cerebral cortex. And we'll also have a better ability to attack questions such as the one at the bottom of the screen, which asks us to identify where an image will be processed in the brain based off of the characteristics of the image, as well as where the image was presented in front of the eye. Let's begin by dissecting the anatomy of the eye. At the front of the eye, there's the cornea, which is essentially a clear, dome-like structure that allows light to enter into the eye. Once the light enters, it'll pass through the pupil, which is a gap inside the iris. The iris is the colored portion of the eye, and with its contractile muscles, it can regulate the size of the pupil. So as the iris becomes smaller, the pupil will become larger and vice versa. So these two have an inverse relationship according to their size. And the iris changes the size of the pupil in response to the amount of light available in our environment. For example, when we're exposed to bright light, the pupil will constrict in order to make itself smaller and protect the retina from damage by overexposure to light. Conversely, when we are placed in a dark environment or at nighttime, the pupil will dilate or become bigger so that we can acquire more light from our surroundings and create an image at the back of the retina. Behind the iris is the lens, and the lens is what focuses light so that it creates a focal point at the back of the eye called the fovea. And yes, this should remind you of optics, which is tested on the physics portion of the MCAT. Keeping in the theme of studying structures at the back of the eye, we have the retina. And the retina is what translates the photons in light into an electrical impulse that can be transmitted to the rest of our body through our neurons. Once the retina detects the image, the information will travel down the optic nerve to be deciphered by our brain. In order to detect light, the retina has two specialized cell types, rods and cones, and these are the light absorbing cells of the retina, and they're able to translate light energy or photons into an electrical signal that can be carried to the brain. Both of these cell types are concentrated in the retina, and remember that light will pass through the pupil and the lens and hit the back of the eye where these rods and cones are concentrated. The orientation of rods and cones in the retina is a little counterintuitive, and the light absorbing portion of the photoreceptor actually faces towards the back of the retina. And this will make a lot more sense by the end of this review. Even though rods are highly specialized, they still have the organelles that we would expect to see in any other cell type, such as mitochondria and a nucleus. However, they do have a specialized synapse, which is oriented towards the front of the eye so that they can transduce that electrical signal to other cell types that will carry that signal to the optic nerve. Rods contain only a single rhodopsin pigment, and for this reason they have a low sensitivity to detail, but rods are better suited for reduced lighting and allow us to have night vision. And since there's only one rhodopsin pigment, rods are not able to detect color. However, cones have a similar structure to rods. They do have a shorter membrane shelf, but they still have the common organelles we would expect to see in any other cell type, and the membrane shells are oriented towards the back of the eye while the synapse is oriented towards the front. Cones have three different types of rhodopsin pigment, and for this reason are able to detect color as well as detail. The three rhodopsin pigments are able to absorb light in the red, green, and blue color spectrums. These are best in bright light, and we see a concentration of cones at the fovea, which is the focal point that lies behind the lens at the back of the retina. Now that light has been absorbed and translated into an electrical impulse, we can see which other cell types it will have to pass through in the retina in order to reach the optic nerve. This is a cross-section of a retina, and notice that the photoreceptors, or the rods and cones, are oriented away from the front of the eye. And this is so that as light enters the eye and passes through the layers of the retina, it will collide with the back of the retina where it can be absorbed by rhodopsin. Also note that this cross-section shows that there's only one cone cell but several rod cells, and this is accurate because there's actually 20 times more rod cells than cones in the retina, with the exception of the fovea. Remember that the fovea was the focal point of light behind the lens, and it's almost exclusively made up of cone cells. Now that light has been absorbed, it can be synapsed onto the other parts of the retina. First, it will be transmitted to the bipolar cells, and these are able to highlight gradients and will pass the signal along to the ganglion cells. However, the bipolar cells are affected by two ancillary cell types, the horizontal and amacrine cells, which both work together to take information from multiple retinal cells and fine-tune our visual image in order to detect edges and contrasts. Now that the signal has been refined, it can be passed on to the ganglion cells, again receiving information from multiple photoreceptors, and these ganglion cells will group together to form the optic nerve. Now that our visual information has finally reached the optic nerve, we can understand the visual pathway, which is the anatomical connection between our eyes and our brain that allows the flow of information. Here is a representation of an object sitting in front of our eyes. Note that each object is split into two halves, the blue half representing the right visual field and the black half representing the left visual field. As light leaves that object, it will pass through our lens and it will cross so that the right visual field will be perceived by the left half of the retina, and the light leaving the left visual field will be perceived by the right half of our retina. Once those signals are absorbed, they will pass through two fibers. The temporal fibers leave our eye on the lateral side of our head, and they're aptly named because they are in close proximity to our temples. And the other set of fibers are the nasal fibers, which are in close proximity to our nose. As this information travels down the optic nerve, it will reach a site called the optic chiasm, where the nasal fibers from the left and right eye will cross. Visual information absorbed by the left side of the retina in each eye will be transmitted to the left hemisphere of the brain, and the visual information from the right side of the retina in each eye will be transmitted to the right hemisphere of the brain. And our visual information will be sent to the lateral geniculate nucleus, as well as the visual cortex and the superior colliculus. Note that because of the crossing in the optic chiasm, we can say that everything in our right visual field will be processed by the left hemisphere of our brain, and everything that was presented in the left visual field will be processed by the right hemisphere of our brain. Well, we finally transmitted all of our visual information to the brain. What are we going to do with this information? Well, we're going to process it, and this is done at three different sites. One being the lateral geniculate nucleus, which is located in the thalamus, the visual cortex, which is part of the occipital lobe, and the superior colliculus. Now, it's not really important to know what each of these sites do for test day. Just recognize that our visual information is processed at these three sites. There are two types of processing that we'll want to know for the MCAT. One is parallel processing. This is where we identify key characteristics in an image and apply those observations to a memory in order to recall a word or a title for the object in that image. For example, when we look at this picture, how do we identify what's really in this image? Well, we identify the key characteristics, some important things like color and shape, and then we say, what does this remind me of? Oh, I remember watching the Discovery Channel and learning that this looks like a cheetah. So we applied a memory to those visual characteristics in order to recall the name of that object. The other type of processing that we'll want to know for the MCAT is called feature detection, and this is when we use a key characteristic to help us filter out important information from that of little value in our visual field. For example, let's say we're going to a concert and we want to find our friend Bob. Well, how are we going to find Bob in this huge visual field full of stimuli? There's lots of hats and colors, and it's almost an overstimulation. There's so much information to filter here. Well, we're going to have to find a key characteristic to help us find Bob. For example, Bob told me last night that he was going to go to the concert rocking a green shirt. So let's use the important information, like a specific color, in order to filter out all the unimportant information in this visual field and find our friend Bob. Oh, there he is. We found Bob. All is right in the world because feature detection helped us to find our friend. Now that we've completed this review, we can evaluate questions such as this one, which asks us to predict how our eye's behavior will change in response to the availability of light. And the correct answer is B. When we're placed in a dark room, the iris will contract, allowing the pupil to enlarge. Remember that the iris and pupil have an inverse relationship when it comes to size. We can also attack questions such as this one, which asks us to identify the visual pathway between the eye and the brain. And the correct answer is A. The crossover of nasal fibers occurs at the optic chiasm. We can also evaluate questions which ask us to identify which cerebral hemisphere will process an image, such as this question. And the correct answer is C. Remember, because of the crossing over in the optic chiasm, everything shown in the left visual field will be processed by the right hemisphere of the brain, and everything in the right visual field will be processed by the left hemisphere of the brain. Therefore, if an object is placed in front of the right eye, half of that visual field will be processed by the left hemisphere of the brain, and the other half of the visual field will be processed by the right hemisphere of the brain. So let's highlight the key takeaways that we want to remember for test day. Once we receive visual stimuli from our environment, it will be focused through the anatomy of our eye and then translated into an electrical signal by the photoreceptors in the retina, the photoreceptors being our rods and our cones. Once that electrical signal has been made, it will pass through the other cells in the retina layer and then be sent to the ganglion cells and transduced onto the optic nerve. The optic nerve will then take that signal to the optic chiasm and other regions of the brain for processing. There are two types of visual processing that we'll want to know for test day. Parallel processing being our ability to recognize key characteristics in an object and applying those characteristics to a memory in order to recall a word to describe that object. There's also feature detection, where we use key characteristics to help us filter out unimportant information in order to recognize our desired object. Well, congratulations on completing this review. I hope you enjoyed taking the opportunity to understand the dynamics behind how our eye anatomy absorbs light photons and how our brain will process that visual stimuli so that we can perform basic tasks like finding our friend Bob. Thank you for joining me, and as always, best of luck on your MCAT.